Well, thanks so much, Eric, for asking me to join. Snapshot Day is something that I have deep in my heart for water quality, and sharing it with everybody is always such a pleasure. So, and I always am excited that perhaps everybody will do a little Snapshot Day at some point during the summer. But our Snapshot Day for 2017 was on May 6th, and this was our 18th year that we were able to conduct Snapshot Day. So it's a long-term program that we've been doing in the Central Coast of California with our partners. In 2003, we actually were able to do a statewide Snapshot Day, but because of budgets, we haven't been able to do that again, but we're always looking for partners to hopefully do that. So maybe somebody in this group will be interested. But just going forward, I work for the National Marine Sanctuary System. I'm here in the Monterey Bay area, but there's lots of national marine sanctuaries all around the U.S. and out into some of our territorial waters. But the Monterey Bay National Marine Sanctuary is here in the Central Coast of California. Here we are in Monterey, which is where our main office is. The sanctuary extends all the way up to Marin County and all the way down to Cambria down south. So we have lots and lots of watersheds that are flowing into the sanctuary, and our idea behind monitoring for Snapshot Day is to try and get an idea of what is the water quality flowing from those major watersheds into the sanctuary. And we use citizen scientists to do that with us. As I mentioned, we've been going since 2000. We want to know what the quality of water flowing into the sanctuary is. Our ideas have been to develop a long-term data set of really good quality monitoring results that enable some resource managers to make good decisions to protect the environment. The Snapshot Day data that we have has been used by the state for 303D listing, and has also been used by some of our other local resource managers to try and identify sources of contaminants or pollution. And of course, our last goal is really to inspire stewardship. Snapshot Day, like other citizen science monitoring programs that we run here at the sanctuary, really help to inspire and inform our volunteers that then sort of disseminate information to their friends and family, and so it's sort of, as word spreads, you get more and more people involved. But Snapshot Day is unique because it is such a short time commitment that for the majority of folks, they can make a huge contribution without having to put in hours and hours and hours. So we have a 2 to 3 hour training period on the weekend before Snapshot Day is conducted, and then we have about a 4 to 5 hour actual commitment on the actual day. So it's about a day's worth of time, and for a lot of folks, they don't want to necessarily put more time into citizen science than that, and this is great. I always call this the gateway for water quality monitoring, because it's a great way to sort of introduce the ideas, give people some experience, and then also help them understand sort of their watersheds. Partnerships are so important to making Snapshot Day a success. Our primary partnership is the Coastal Watershed Council, and they're up in Santa Cruz. And they run the Santa Cruz Hub, which is the gathering spot for Santa Cruz County volunteers. And they manage those Santa Cruz County sites that we have. We also work with the San Mateo County Resource Conservation District. They also help to organize the hub and bring in the samples and get the samples to the different labs that they need to go to. And then we also have lots of other folks, the Sewer Authority, the County of Santa Cruz. Cities also help us quite a bit. This year we had a lot of extra funding from our local cities. We use county health labs. And then we also use other federal agencies and then also local nonprofits. So it takes a village to make something like Snapshot Day happen. Some years we have lots of partnerships. Some years we have less. This year we were really fortunate to have a lot of people in a lot of different organizations step up and help us with funding and also with recruiting. So it was a fantastic year this year. So our stats from 2017, we had 146 sites that we did, and this map here is from 2016, so it's last year's map. I don't have this year's map yet. We had a number of sites that were up in the Salinas Valley that we were able to monitor this year because we had so much flow in past years because of the drought. We just haven't had enough flow, and so we've not been able to monitor. But this year we were able, with the help of the Upper Salinas Las Tablas RCD, up in Templeton, we were able to do a number of sites up here in the Upper Salinas Valley. We had 179 volunteers, and that's sanctuary-wide, so that's all the way from Half Moon Bay in San Mateo County down to Cambria in San Luis Obispo County. We have 4 hubs that we have the volunteers meet at. So San Mateo County volunteers all meet up at the San Mateo County hub that's in Half Moon Bay that was run by the San Mateo County RCD. Santa Cruz County volunteers, they all meet at the Santa Cruz hub that was run by the Coastal Watershed Council. The folks in the Monterey County area, they all meet at the hub that we have in Marina. And then our folks down south in Cambria area, they all meet at the Cambria hub. So having the hubs is critical because you need to have somebody there who's sort of overseeing the project, able to take in samples, able to answer questions for volunteers, and make sure that all the samples and all the data that we're looking for have actually been collected once the volunteers and once the teams are returning to the hub. So what we're looking for is a sort of a suite of field measurements: dissolved oxygen, pH, air and water temperature, conductivity, and then transparency and or turbidity. Most of our volunteers are doing transparency at this point. And then we collect samples that are lab tested for nitrate, orthophosphate, E. coli, and total coliform. Those are just our very basic water quality parameters that help us to get what we call a snapshot of water quality for our local areas. And here you see 2 images. The top image is from the Carmel River watershed, which is where a lot of for the Monterey Peninsula, the drinking water is taken from. And so people are very concerned about water quality in that watershed. And then the bottom image is actually from a heavily ag area. And so, and those tend to be our most difficult spots. So, let's see. Okay. So lessons learned about Snapshot Day. You know, training the volunteers is the most critical part of the whole event. Making sure the volunteers understand step by step how to do everything, how to perform the data collection, how to perform the sample collection, all that. Having really detailed but not too detailed descriptions of how to take conductivity or how to do the dissolved oxygen is really important. The training is critical for the volunteers, and I can't say that enough because when you have volunteers that aren't trained properly, something happens out in the field and then everybody comes back and they don't have samples or they don't have the information that you're looking for. So training those volunteers is pretty critical. And I always like to say, you know, if you can't explain it to a 6-year-old, just like Albert Einstein, then you don't understand it yourself. So it's really important to help people understand why something like transparency is important or why collecting samples is important in a particular way. So I always have very detailed training sessions on how to fill out the data sheets and what it is that I want folks to do. The volunteers go out with empty data sheets, but then they also have a filled-in data sheet so that they can look back and say, okay, we're missing this information for Lisa and trying to fill in the data sheets properly. Recruiting from local colleges has proven to be just an amazing place to find volunteers for me. You always have to provide food for the college students because they run on their stomachs, absolutely. But the local colleges have been so important for our Snapshot Day volunteer recruitment. The volunteers that are there are eager, they're interested, and they don't necessarily have a lot of experience in doing any kind of field work. This is a great way to develop a broader volunteer core but they also, because they don't have as much experience, they don't have preconceived ideas about what should be happening, and so they tend to follow your directions a little bit better. Sometimes the more difficult volunteers are the ones that actually have the most experience, and they only do what they've been trained to do from previous events. And with college students, they don't have that same experience, so they actually follow directions really, really well. So our local students have just been amazing, and I always like to encourage them to get involved in our other monitoring events because I always wonder, what is going to be that thing that's going to spark their interest and their passion and possibly make them interested in the field of water quality and monitoring in general. So, one of the things I've learned over the years is to get reliable help at the hub. Everybody that's gone through training, everybody that's an existing volunteer that shows up always wants my time and my attention. And if I'm trying to get people signed up and trying to get people kind of going with their teams and get everybody connected, that can be very, very challenging if I'm being pulled off in all these different directions. So I try and find some existing volunteers to come and help me at the hub. I deputize folks pretty easily and get them to help people fill out paperwork, help to wrangle anybody that we have coming to talk. Sometimes we have dignitaries, and so that's super helpful as well. One of the things that I started a number of years ago when I first was doing the Snapshot Day coordination is I started a photo contest. Snapshot Day, because of the name, it gets folks that are interested in photos, and so we have to sort of explain to them, it's actually about water quality. But I do really enjoy having the photo contest because it's a great way for the volunteers to be creative and to show what they're interested in, but it's also a great way for us as the coordinators to kind of spy in on what's going on while the teams are out in the field, because you don't get that opportunity any other time. In Monterey, we have close to 20 teams that go out to the different areas, and so I couldn't go around to all those 20 teams. They're so far apart. And so having everybody take photos is actually a great way for me to see what's happening, what are things that I might see from a bunch of teams that aren't exactly what I want people to do, and how to address that in the trainings in the future. So, and then we always have best technique. So the lower right image is for our best technique category. Somebody's taking some DO measurements with the Kinets kit. We have a team shot, and then we also have the funniest. And the funniest brings out some of the best, most creative ideas from folks. I always love, in the upper right, there's my team from San Mateo County. Every year they come up with something just absolutely hilarious. That particular year that this image was taken was when Snapshot Day was on Cinco de Mayo. They've done other stuff like trolls. They've all dressed up as trolls and done just crazy things. So I always love it, but it's also sort of secretly my way to spy in and see what's going on. So looking at the data, every year what we do is we put all of our data into a GIS and map out some of the areas that are the highest concentration of analytes. And so this is one of our maps from 2016. I don't have my maps from 2017 ready yet, so I'm just going to show you guys last year's data. But you can see here in the central part of Monterey Bay, what's at the bottom of really pretty much 100 miles of agriculture are some of our highest areas for nitrate. And here's over 10 milligrams per liter of nitrate as N in this particular zone. So you get a sense of what that's like. Here's for E. coli, similar idea, where at the bottom of this agricultural area and in some of our urbanized areas we have higher E. coli, and also way up here in the upper Salinas area. And then what I do is at the end, after I've looked at all my data, I actually will assign data or assign site areas of concern that exceed a either a field water quality objective or a lab water quality objective. So for 2016, you get an idea of where some of our areas of concern are. And these aren't necessarily a formal designation. It's more of a designation for us and our partners so that we can start looking at some of these areas and try and address some of the issues. So here in the Elkhorn Slough, Morro Coho Slough, Alisal Slough, Salinas River area, there's usually a lot of areas of concern in there. And then in 2015, we did a 14-year report. It's on our website. In that 14 years, we had almost 2,500 volunteers that came to participate. We monitored about 243 sites. There were 186 sites that we had monitored over 5 years in the 9 major hydrologic units. And what we did is we looked at exceedences and trends. So here you can see these graphs, these bar graphs of exceedences by hydrologic unit. Estrella is not listed on our bar graphs because it just didn't have, it's so infrequently flowing that we didn't have much data for it. But you get a sense of, in the Salinas watershed, there's more exceedences for nitrate than, say, in the Big Basin watershed. So, and that's all in our report. That's available online. Then we also looked at trends. So here you can see for Tembladero, which is again in one of the bottoms of the watersheds where there's a lot of ag, and then also for Elkhorn Slough, the nitrate along there as well. So those are all things that are available online in our report. So if you're interested in any of the information, or if you want to try and start up a Snapshot Day type event, I'm always willing to talk to folks and help you get started and help you figure out areas to sample, or just give you insight into kind of what we have done in the past. And as I mentioned, in 2003, we did coordinate a statewide Snapshot Day, and we would love to try and figure out a way to get funding to do that again. It's been more than 10 years since that event, and it would be really wonderful to figure out a way to do that again. So, and then our 14-year report is available on our website, which is montereybay.noaa.gov.
All right. Thank you so much, Eric. Really appreciate it. Hi, everyone. My name is Savannah Rudraff, and I'm the Natural Resources Associate at the League to Save Lake Tahoe. The Tahoe Truckee Snapshot Day has been going on for 17 years now, and this past Tahoe Truckee Snapshot Day was my 3rd year coordinating the South Shore event. So as I mentioned, I work for the League to Save Lake Tahoe. We're better known as Keep Tahoe Blue, and we took over coordinating the Snapshot Day event about 4 years ago. We focus on Tahoe's clarity, and we do that through 4 ways. We combat pollution, promote restoration, protect the shoreline, and tackle aquatic invasive species. You can learn more about our nonprofit at keeptahoeblue.org. So for Snapshot Day, we go throughout the Tahoe Truckee region, so that is the entire Truckee River watershed, but Lake Tahoe is the most popular sub-watershed within the Truckee River. So we have 4 locations throughout, from the Truckee River headwaters in Meese Meadows to Pyramid Lake, and it covers approximately 150 miles of river. The 4 locations that we have within that is the Reno location, and they have school groups go out from Reno all the way up to Pyramid Lake to monitor streams and the Truckee River. Then we also have a location in Truckee. That one is based in California. The first one is in just Nevada. And then the North Lake Tahoe region is in Nevada and California. And then the League focuses on the South Lake Tahoe region, which is also in California and Nevada. Throughout these 4 regions, we monitor approximately 82 sites per year, and we have 417 volunteers who came out to join us in 2017. As I was mentioning, we focus on the South Shore region of Lake Tahoe, And we monitor approximately 32 sites from Meeks Bay to Zephyr Cove. And we pick these sites based on the number of years that they've been monitored in the past, that we can get consistent data. We also bring in new sites based on projects that are happening within the Lake Tahoe Basin at the time. And we also get rid of sites during drought years when they are no longer flowing. This year we didn't seem to have that problem. All of our sites are still very much so flowing with the snowpack we still have up in the mountains here. In 2017, we had 111 volunteers join the Snapshot Day event. And with those volunteers, we want to make sure that we are promoting environmental education to the school groups that we bring out and instilling a sense of connection and stewardship within our volunteers. Many of our volunteers have never had the opportunity to do field work like this, and so they really get the chance to learn a lot through this experience, while also collecting valuable water quality info, which includes screening for any large-scale water quality issues and providing a snapshot in time of watershed health for people who are looking for that data for restoration projects or what have you. We also want to make sure that our volunteers are having lots of fun and feeling very appreciated and feeling a sense of giving back. This volunteer is having a great time holding a thermometer in the water just outside the Tahoe Keys area in a waterway going into the lake from these lagoons. The data collected is then compiled into a database that is made available publicly online, and you can check out that database at on our website at keeptahoeblue.org. We also have annual reports for all 4 regions that are made available at tahoetruckiesnapshotday.org. And then we use this data to create visual depictions so that it is easily seen, the differences between years. So the first map on the left is from 2014, and then the map on the right is 2015. And these two maps are depicting the fecal coliform levels in the water. The color is appropriate to the levels of fecal coliform within the sub-watersheds there. And so we have maps like this for every constituent that we are measuring. In order to assure quality data, we first put team leaders through a training that lasts approximately 2 hours. And this training starts out in the classroom, and we have a presentation talking about all of the words that they're going to encounter that they may never have encountered before. And then we go out into the field in order to teach them how to use the instruments that they will be given. Here in this picture, I am teaching a team leader how to use a dissolved oxygen kit. And then we also want to recruit team leaders back year after year, and preferably to the same sites that they have been monitoring in the past, so that they are familiar with each site. They know exactly where to sample each year, even when it's drought conditions or flooding conditions, like the flooding conditions seen in this picture. On the actual day, volunteers show up to Lake Tahoe Community College, and they check in before sitting through a presentation that briefly describes what they're going to be doing out in the field and why it's important. That the team leaders can educate the team members even more since they've been through extensive training. And so then we want to get the volunteers into the field as soon as possible, because that is why they're there, and they want to go out and have fun. So the volunteers then split up into groups with their assigned team leaders, and go out to their respective sites. And they do all of the water quality monitoring with the equipment that they've been given. The team in this picture, they are measuring for phosphates with a Lamont water quality testing kit. This is merely for education of the volunteers because they also take water samples that are then analyzed in the lab. But this gives them a chance to actually learn more about phosphate presence and have a real connection there rather than just turning in samples to a lab and possibly not seeing that data again if they don't click through on the link that I send them, or maybe that data won't mean anything to them by the time that it comes, because we just got that data back a couple days ago. And the event was a month ago. So each team is given a field data sheet, and they go out and fill this out first off with the site name, station ID, and the team leader name and phone number, just in case I have any questions upon submission of the data, and then the team members' names as well. They then complete an observation section where they can describe cloud cover, any wind, water clarity, etc. And they also let me know if there has been rain in the past 24 hours. Of course, we want them to give the precipitation amounts, and so we post this at the staging site that morning so that everybody gets the same amount of precipitation, and when the data is looked back on later, it can be accurate. And then they get into their field measurements, and they let me know the instrument ID, the time of collection, and then the first result and the replicate. We give them a range to expect so that if it's outside that range, they know to for sure take a replicate or even take another as well. As you can see here, they go for many different parameters. For pH meters, for pH, they either use a meter or a strip. Dissolved oxygen, we provide those kits that was pictured previously. And for total dissolved solids, or TDS, we provide TDS meters. The nitrate and phosphate here is for the Lamont water quality test kit. And this is only given to certain sites based on their location and the amount of time that that site may have after taking all the other parameters. Down here they have their lab samples that they collect, and so they have bottles to collect for nutrients, and then the top water of one of the bottles is taken off to analyze for turbidity right there at the staging location when they get back and turn in their samples. We also provide whirl packs for fecal coliform bacteria samples. Then they complete a visual assessment, and each team is given visual assessment forms, one for each site, just as they are given the field data sheet. And then the visual assessment form encourages them to take a team picture. It encourages them to take pictures of the substrate, upstream and downstream, and then they walk approximately 100 meters upstream from their sampling location to look for any input, any erosion, or anything else that may influence the sample that they took. For our Snapshot Day, we want to get a lot of volunteers of all ages and backgrounds out into the field in order to experience this. And so for that reason, we partner with the Boys and Girls Club of Lake Tahoe. We partner with TRIO, which is an Upward Bound program here in Lake Tahoe. We actually partner with UC Davis, freshman biology. And so we have approximately 30 UC Davis students come out each year. And this year they actually did bioacoustic monitoring as well, which was kind of a pilot for our Snapshot Day. We also partner with Lake Tahoe Community College, so we get lots of local community college students out into the field in order to get practical hands-on experience, as we have a very large environmental education program here in Lake Tahoe. And then we also get Seroptimus International involved as well, and that brings, those women are so enthusiastic about the event that they bring a lot of their friends as well. In addition to that, we have the South Tahoe Public Utility District and Lahontan Regional Water Quality Control Board as our two laboratories that are analyzing the water samples that are brought in. And then we partner with local businesses for donations in order to show our volunteers our appreciation for their hard work. We get pizza donated, we get coffee and pastries donated as well, and some cookies for dessert after the pizza. And this year we had fruit, which was a nice healthy option as well. So that is our Snapshot Day, and if you would like to reach out and learn more, as I mentioned quite a few times, you can visit keeptahoeblue.org in order to view our data set, or you can reach out to me directly at my email, savannah@keeptahoeblue.org, and I would love to answer any additional questions that you may have after today. Thank you, everyone.
All right. Well, thanks everybody for the opportunity to talk a little bit about Snapshot Day. I was lucky enough to be one of the team leaders down here in Southern California back at the 2003 statewide Snapshot Day that kind of got a lot of this going, and it really set the framework for us to do a lot of different type of Snapshot Day work. So that's what I want to kind of cover the big picture of the different types of Snapshot Day events that we've done and the different elements of those. So we've done a wide variety of Snapshot Day events. They have been our biggest one, covered all of Orange County. We had about 28 sites and involved about 12 different organizations participating. So it's a lot smaller than you know the two groups that presented before that have really large programs. But it was pretty significant in the realm of Snapshot Days down here where we are. And then we've done really small ones where we involved just basically a high school group or something like that wanted to collect some data on their local water body, wanted to involve students and things like that. So that's where I get to the first part. The first element there is the purpose. Is for us collecting quality water, quality data is always an important aspect. So that's something that we always have as a goal. But there's different levels of how we're going to be able to do that. For instance, in some of our larger events, we had teams with different skill sets. Some of the teams would use really high quality equipment, had been done water monitoring for years, and we would send them out to specific places to collect really high quality and lots of water quality data. Other teams, we would be using things such as color comparators and other things like that still collect, usable usable water quality data that meets QAP standards and stuff like that. And then we would have other teams which may be just some scouting groups or church groups or just community groups that would use cup kits, and they would collect basic data depending upon what they were able to do. So the idea was, in some cases, it was more educational and less geared towards super accurate readings. On the other hand, some of it was going to be very accurate, super high quality data, but we made sure all of it had some kind of a use. So for instance, even if somebody was using a cup kit or something like that, if they went out and they came back with some kind of a really bizarre number or something that looked realistically but was kind of a little scary, then we would back that up by sending another team out to go out and follow up that monitoring and find out if there was a real issue. The second thing I have here is funding. Funding's absolutely critical. So, you know, if you're going to try to do something on a large scale, it's going to take a significant amount of funds. And you know, I really commend the groups that have been doing these really large scale ones for many years. I know how difficult that is. And, you know, the key things that got talked about were partnerships and things like that. You absolutely can't do it yourself. You absolutely can't fund it yourself. I've had a wide variety of funders for different reasons. I've had everything from corporate sponsors who pitched in because they were going to have some of their employees come out and participate in the event. I had the county participate. It was part of their MS4 permit outreach. They simply needed to get water quality on the minds of a certain number of people, and this was another way to do it. And then we've just had individuals or other funders, foundations, etc., pitch in at various times. So there's really a wide variety of funding available out there. So be really open-minded on that. Don't think that the car dealer in your town may not want to pitch in for something like that if they can get their name on a logo, or more importantly, if their employees or something can participate. On the planning side, one of the things that you really want to plan for is access. I know for our area, we have a lot of our creeks are considered flood control channels, and they're fenced off. They're vertical walled concrete channels. They can be difficult to get to. So you want to identify places that people can get to, and then places of significance. So a lot of the monitoring that we've done, we focused on creeks that empty into marine protected areas, so that we can get information that would be related to marine protected areas. We also done monitoring of creeks that empty into ecological reserves, things like that. And then also, occasionally, like I said, when there will be a school group or something like that, or even a city that says, you know, we don't really know what the water quality is in our park lake or something like that, and that's important to them, so that's a good opportunity to do that. As an example of one of the things we did, we sampled all of the park lakes in the city of Huntington Beach, along with a creek running down to the ecological reserve, so that we had a lot of varying different data, and it was easy for people to access that. Getting to the city park lakes, for example, was something that was easy and safe for people to do. And then also they really felt connected to their neighborhood. We had a number of parks so people could go to some place close to them. So it wasn't some abstract thing like, well, what's the water quality, you know, in the ocean that maybe I don't go to, but you know, that lake and that park is right around the corner, and that's something I'm concerned about. Promotion. It's really important to promote these events through every possible aspect you can think of. So if you can get it in your obviously, you know, there's social media and all that type of thing, which is good for getting the word out. But you know, voice to voice, getting people to talk about it in your partner groups, all that type of stuff. That one-on-one promotion really helps turning out people. For me, a big Snapshot Day event would be if I got 100 people to participate. So it's not something that draws real masses of people. We do trash pickups, for instance, that have 600 people show up at a time with very little promotion. These types of events are a little bit different. People think that water monitoring is hard. They think that they may not be able to do it. So you need to make it clear that it's really easy. And then the last thing is your results. We always, no matter whether it's a small little event or a larger one, produce a report of the results. And then we try to report those out to whoever the local officials are, or whoever the user group is. If it's a county park, if it's a city park, we try to get that information to the people at those places, so that they know that this is happening, that this data is available, and that, you know, then we offer to work with them in the future if we identify any problems, or also just if it's something that they want to do again to gather more information or to involve the public. So that's my short spiel on just kind of the grand scheme of Snapshot Day events, and I'm glad to talk to anybody about any thing that you may be thinking of doing, or different strategies or things like that. So this is my contact information, and with that, I'll turn it back over to Eric to open up for questions.
So let's take some questions. These were great presentations. It looked like a lot of the sites that were being monitored in the 3 different programs were integrator sites, that they were at the bottom of particular watersheds or drainage. Lisa, would you like to talk about the value of that, why you were selecting sites that were at the bottom of a watershed?
Sure. We select a lot of our sites at the bottoms of watersheds because then you're sort of collecting all of the pollutants or water quality from all of the sites above. But a number of our sites on some of our watersheds, they actually are monitoring a number of points. Like the Salinas River is over 100 miles long. And we actually monitor a number of sites along there. My 2016 map didn't have those sites on it because we didn't have money to monitor those, but in 2017 we were able to do it. So for some of the larger watersheds, we monitor a number of spots along there so we can sort of bracket some of the different uses that are there. But due to funds and also size, a lot of our watersheds are rather small, so we end up just monitoring at the very bottom. You wouldn't want to monitor at the top without doing the bottom because that wouldn't tell you a whole lot about what those uses are in between. So we monitor at the bottom so we can sort of measure all those different uses that are upstream.
Thank you. Yeah, I also noticed that there was involvement in a lot of the programs where there was really good representation of the public. Environmental justice communities were able to participate. Wasn't mentioned, but I know that tribes have been involved in some of the snapshots, especially at Pyramid Lake. And then the diversity of people in the pictures of your college students and young people. How have you been able to accomplish that kind of outreach amongst your snapshot days? If all of you could chime in on that, I'm sure everybody would like to hear how you were able to achieve that kind of diversity.
Well, I can go ahead and start. This is Savannah. Our Seroptimus International, amongst other groups like that, they actually recommend groups they've worked with in the past in order for us to involve people of many different backgrounds as well as many different ages. It's also, most of the time when you reach out to those different groups, they're very, very willing to participate, and they really want the opportunity to get involved, whether it's a few of them or if it is 50 of them. And so we've just really played on our connections, and we've really reached out quite a bit to make new connections. And it doesn't always work out, but it has worked out enough that we've been able to reach out for
This is Ray. So one of the strategies that we use are reaching out to groups that have really wide bases. So, for instance, Boys and Girl Scouts. I said for high-level monitoring, that's not what they're interested in. But if you're having them use color comparators and other things, Boy and Girl Scout troops are everywhere. And so if you've got a diverse area like Orange County that's a lot different inland than it is along the coastal areas, by working with the scouting groups and other kind of groups with large ranges, you can get that wider diversity of people.
Yeah, this is Lisa. I'll second what Ray and Savannah have said, and that just trying to find folks to reach out to. People are really interested. Everybody's, oftentimes have questions, especially for our local area when they go to the beach and they see that the beaches might have been posted for bacteria. There's all sorts of, different groups out there, and it's just really a matter of trying to make connections with them. And sometimes for myself, I'm able to go in person. Other times, I just try and keep everybody kind of going over email. But yeah, and every year our list of participants or interested folks seems to grow. They're not always able to join every year, but we're always looking for new groups, and so that's a lot of what kind of coordinating an event like this is sort of rallying the troops and finding people to join, and so that takes a fair amount of time.
The data that was presented on the maps, we saw some exceedences. When you find those exceedences of a water quality objective, how is that being treated? Are you guys being able to go out and do the follow-up work? Ray mentioned, that's been done for, they see an exceedance or a problem on a lower quality monitoring kit that they do some follow-up. But what's been happening beyond that? I'm interested in data sharing with agencies and, potential finding of pollution, illegal hookups, or what's been happening within your snapshots over the years as far as one day going out in the field and identifying a problem that is then being looked at and corrected over time?
Well, this is Lisa. I'll say for us, we look at the areas of concern that we sort of designate from the data that we have. We also share our data with the state board through the Exceeden data portal. And then we also share a lot of our data with a couple local NGOs that are focused on water quality monitoring in specific areas. So there's always a local group of folks that are interested in it, and then on up to the state board. And yeah, it's an issue for us when we see something that's consistently over water quality objectives. It's an issue for us to try and find money to deal with certain areas or certain stretches, and that's always going to be something that we struggle with. So we're always looking for more money to try and improve water quality. Absolutely.
This is Savannah. So when we find those issues, as a small nonprofit, we don't have much capacity in order to follow up with it ourselves, but we use that data and alert our partners, such as Lahontan Regional Water Quality Control Board. And we are fortunate enough to be in an area that has very stringent water quality rules, and so a lot of times what it just takes is knowledge of the issue and then enforcement of those rules that are already in place.
And then this is Ray. Don't underestimate the idea of doing a media release on your results. So we've done that on several occasions, and we've had a couple spots where we've documented problems year after year. And we've actually used media releases on our results from our Snapshot Day events to highlight that. And then, and then we always give the data, our report, to any area, that's interested. I mean, we try to give it to everywhere that where we monitor.